This is the Angry Birds Bird Minute, sponsored by Angry Birds for their 15th anniversary, where we tell you all about a cool bird that may or may not be angry. Today's bird is the oxpecker. Oxpeckers are named for their lifestyle riding atop large mammals like cattle, zebras, wildebeest, giraffes, take your pick. They mostly eat invertebrate parasites, which accumulate on their steeds, but can occasionally take a little bite of the mammals themselves. They roost in vegetation, but sometimes might also grab some Z's without leaving their host by hanging under a giraffe's thighs or atop a buffalo's back. But are they angry? On a scale of 1 to angry, we rate the oxpecker as tick old. This is Wild Green Streams. I'm Rhett, and today's guest is Justin Stewart. Justin is an evolutionary biologist working to understand the relationships between plants and fungi. Justin, welcome to the pod.
Thank you. I'm very happy to be here and talk about fungi.
So, what are mycorrhizal fungi?
Mycorrhizal fungi are a special, very ancient group of fungi. So they've evolved multiple times across the tree of life. So it's not like they're apes where they had to evolve once, but they've popped up many different times over around 450 million years. They're these fungi that form really intimate relationships. So they penetrate plant roots or they live on the outside of them, and they take carbon the plant gets through photosynthesis. And in exchange, these fungi grow these vast, extensive networks. You can think of it as like these highway systems underground that take nutrients and trade it. So there are these microbes. We call them microbes or microorganisms, and they live with plants in symbiosis with plants trading nutrients for carbon.
They kind of take the form of networks, right? Like to look at this thing, what does it look like?
This is why I'm so excited to be able to talk about fungi. There is no charismatic megafauna. There's no polar bear on an iceberg that exists for fungi. But so they take a few different forms. There's many different flavors or varieties or tastes of mycorrhizal fungi. The one I work on is called arbuscular mycorrhizal fungi. And these make these vast networks underground that maybe are, 3 micrometers wide, so much thinner than a hair. But then they can be maybe, 5 to 10 meters long in a cubic centimeter of soil. So it's these thin threads that are wrapping around. Then you have other ones that actually do make mushrooms. So the ones I just told you about never make a mushroom. But if you've ever eaten a carrot, you've probably eaten this fungus, but not known it.
Everyone just threw their carrots away.
Right. No. Oh, I hope you know this is not like The Last of Us or like cordyceps kinds of thing. These fungi, I think, are very beautiful because they're very sophisticated for being what looks like either a mushroom in the forest or maybe just a tube underground. They do very, very sophisticated behaviors and are critical to all life on Earth.
So, what are they doing?
Right. So these fungi, I said they're really ancient. So if we go back in time, they're about 10 million years older than plant roots.
Mm-hmm.
So that's 10 million years older than plant roots. Like roots are the new guy on the scene. So the main thing that these organisms are doing, at least from the human-centric view, which often relies on thinking what do plants do, is they extract these nutrients in the soil and trade them in exchange for carbon, and they grow these networks. They do this in a very, very special way where they actually are connected to multiple plants at once. So they've had to figure out over evolutionary time how to evolve trading strategies and strike bargains and make sure they get good deals on their investment. So if you imagine that you're this network of fungi in soils and you're connected to multiple plants, maybe some grasses, some trees, and maybe some herbs, how do you decide who to invest the most resources into? Who are you going to build the most network for and trade the most nutrients to? How do you evaluate your traders? But at the same time, these networks are vast. The current estimate is that they're, if you took all of them and stretched it end to end, it's about half the length or the length of the Milky Way galaxy.
In the world or just
in the world, and maybe the top 10 centimeters of soil. I'm working on a project now trying to actually map this. But the thing is to think about that if these are a conduit, a connector between this above ground carbon above ground plants, and it's a way to get carbon below ground. They're physical infrastructure for pulling carbon below ground. So I'm very excited to think about these in the context of climate change because it's
Hmm.
infrastructure that's already built there. It's been working for 450 million years, and we're just starting to understand how these organisms decide to cooperate with different plants.
Are these things like a brain? I'm trying to think of a way to ask this question that isn't too deep. How do they process information?
This is a very exciting time to be studying microRIC because we're just like at this cusp of being able to have tools to answer questions about how fungi process information. We're able to integrate high dimensional large data sets using like AI and machine learning, for example.
Hmm.
In our group in Amsterdam, we work on imaging these fungal networks and tracking carbon particles to understand how trade decisions are made of where specific particles of lipids go. Lipids are made of carbon. Um, so that's one way we try to figure out how are they making decisions, or are they making decisions? You know, there's not really a brain in these organisms. It's very
Right.
easy to what is the word people say?
Anthropomorphize?
Fungi. But that's a great question. So imagine that you're a fungal network in the soil and you're connected to three different plants. But in reality, you're maybe connected to dozens. But let's just make it three for simplicity.
Sure.
How do you make a decision? Do you have to evaluate across your entire network architecture, or can you do you have to make these global decisions where the whole network agrees, or are local decisions also a possibility? And those are things that we don't have answers to at the moment.
Kind of like trying to figure out how aliens think.
I don't know. It's such an interesting way that humans talk about this. So we talk with this human-centric above ground bias,
Yeah.
but these organisms are older than us and they've done sophisticated behaviors much longer than we have been alive.
Right.
Yet we have to say they're aliens when we showed up on Earth
That's a
hundreds of
great
millions of
point.
years later. Who's the real alien? I think it's probably us.
Oh, by far.
But that's
Yeah.
it, right? It's just a completely other way of processing information. But it's exciting because this is that time where we can start to use things like network analyses and graph theory and complex mathematics at scale to try to understand how these organisms process information.
So you said that they cover the whole world basically, and it's about 10 centimeters deep in the soil. How much have we mapped these networks?
When we say map these networks, it's very difficult to describe what mapping them is. So one of the large projects I work on at the Society for the Protection of Underground Networks, it's called SPUN, is we're a nonprofit. And what we're doing is we're working with scientists all around the world to collect soil samples, integrate these across very diverse data sets, and then create predictive maps of the biodiversity of these fungi. But most of the soils on Earth are unsampled. So to put in context, there's 150 million square kilometers of terrestrial land, of land on Earth.
Mhm.
And I think we can say that less than 10,000 of those have been sampled for mycorrhizal
Wow,
fungi,
that's
which
nothing.
that's true for a lot of organisms, right? We can say
Sure.
that for a lot of things, but these organisms live below ground. So it's not like we can just view them from space with a satellite. We have
Right.
to actually take samples and identify where they are using
Right.
using DNA. We can't see them with our eyes, so we actually have to look at them at their molecular level.
It's amazing how little we know about things once they're underground. I worked on a project about 10 years ago that was trying to learn about pocket gophers, which are this little mammal that, I mean, they live all over, but it was the southeastern pocket gopher I was working on in
Are these the
Florida.
one? These aren't the ones in Colorado, are they?
No, but they're related. So they're similar. They have teeth on the outside of their face because they use it to dig with, even when their mouth is shut, and they have big pockets in their mouth that they carry things around with through their tunnel systems. But it, we were trying to figure out, and this is a vertebrate animal in one of the biggest states in the United States, and what we were trying to figure out is where are they, and how many species are there in Florida? Like basic, like 1800s questions. And that was for a vertebrate animal. So I can't imagine. It must just be totally wide open for a fungus.
But at the end of the day, it's not wide open. It's so difficult, but it's something that's very exciting. I'm always excited. Like I'm finishing my PhD, but what I'm excited about is that this opportunity, like right now is the time we can do it. So, for example, we use these DNA sequences and train AI algorithms on like massive Google servers, and then lets us predict where all these fungi live. But we do that using satellites at the same time. So it's these two completely different scales, individual DNA molecules,
Hmm.
supercomputers, and then satellites all connecting to answer this question.
Right.
maybe we should try gophers next, doing something similar. But,
It might be a little bit easier because they're pretty easy to see.
yeah, it just feels like it's this moment in history that like it's just like the sweet spot. And since they're so integrally linked to things like climate change and the flow of carbon from above ground to below ground,
Right.
it feels like the moment to start working on them.
Sure. And so mycelium are 3 microns thick usually, you said?
Oof, this is hard. So this was a big project I've been working on with people in Amsterdam is trying to understand the biomass of mycorrhizal fungi. So one specific group called AM fungi. Um, and so if you think of it, like imagine you're sitting down in a park and you look up at a tree, and so you're looking at it from below. And if you look at their branches, you have some that are really thick, some that are really thin, right? So the width of these branches varies across the architecture of that tree crown. The
Okay.
same thing happens for fungi. So they vary their architecture, sometimes up to tenfold. So
Mhm.
some parts are very thin, some are very thick. It varies per species, it varies per ecosystem.
Sure.
Sometimes it varies per day when we go look at them, because they grow. So mycelium is a bulk term we use to describe what we call hyphae. So hyphae are the individual threads.
Sure.
And if you think of clumps of hyphae, that can be mycelium. Then if you think of a mushroom, it's made of mycelium, which are made of clumps of hyphae.
Right. Okay. And so these hyphae are very small, but there are so many of them, and they're so woven together that the organism can get to be like hundreds of tons. Right.
So I can't say the number, but that's one of the projects I've been working on is how much these organisms weigh.
Mhm.
Just like so a recent study that came out of our group, it asked how much carbon goes to these fungi every year. And I'm just going to talk about AM fungi. So again, these are not the
Okay.
ones that form mushrooms and forests. I'm biased. I study the other ones.
Sure.
Um, about 1 billion tons of CO2 equivalents are transferred to AM fungi every year. So that is a very large amount of carbon, but we have no idea how that's actually integrated into the fungi's bodies. So this is many species taking in this carbon. So one of my big questions is how much infrastructure is required to complete that resource transfer? How much fungi do you actually have to accept 1 billion tons of CO2?
Okay.
How many networks can that build?
Have they ever been found caching things? Like, do you think that instead of incorporating it into themselves, sometimes they're just like storing carbon or storing nutrients in little pockets of the soil?
So these mycorrhizal fungi this is another reason why AM fungi, arbuscular mycorrhizal fungi, are hard to study.
Mhm.
They make multiple structures. So they make spores in soils as well, which are like these three-dimensional spheres, sometimes elliptical. What these spores do is they hold massive amounts of lipids. So it's like, think of it as like a seed, right? Like, we eat a seed and it's because they're filled like nuts, they're filled with energy. Same concept with these spores. So they store it there,
Mhm.
but yeah, maybe I can break down the structure of it. So these networks form in soils, and then on these networks, they form these spheres, these spores.
Mhm.
And at the same time, the networks will grow and penetrate a plant root for arbuscular mycorrhizal fungi. They literally go inside of it. It's a very it's like an orchestra of communication between the plants and the fungi, like a molecular orchestra. It goes inside and it creates this thing called an arbuscule, which is like an unfurled tree. And that's like the stock exchange. That's where the bargaining is happening. That is where the fungi are trading and releasing nutrients in exchange for getting carbon. And so these are the major structures. They can also form these things called vesicles inside roots, which are also for storage.
Are they eating their own spores sometimes?
So that's a really great unanswered question. So if you think about a spore, maybe up to 95% of it can be filled with lipids, so carbon. So like
Mhm.
it's filled with that, but it's unclear how this fungi will, in stressful situations, how do they reevaluate their decisions? Do they decide to, and it's also, I say, do they decide? It's unclear who's deciding what, or if this is actually an active decision.
We
But
get the
does
idea.
network architecture, do they grow thinner hyphae? Because then it's less carbon, right? Because the amount of carbon used to build these fungi depends on radius squared, so on its volume, because they're like cylinders. So a thinner hyphae means less carbon used.
Mhm.
Or do they empty spores? But it's very difficult to understand these organisms because they are not like most life on Earth. These fungi are like one large cell, different species, and of course not all networks are connected, but they're one large cell through which there's no cell walls. So carbon and things flow through freely. including nuclei. So there's hundreds of nuclei shuttling into these
Oh,
spores at once. When compared to something like an animal cell or a plant cell, there's one nucleus per cell. So spores can be packed full of carbon and nuclei at the same time. And so we don't know whether or not they break down these nuclei when they need something like phosphorus to trade to a plant host, because nuclei are filled with DNA. DNA is very rich in phosphorus. These are all very open
hmm.
questions.
Wow, that's crazy. Completely different note. Which country that you've been to do you think is the best to do research in, and which do you think had like some unexpected misadventure?
It's hard. I've been to quite a few remote places. I've done a lot of work in the Andes, the Amazon, and the Galapagos, where I was living in Ecuador for a little over 3 months. I guess one of the craziest experiences was we were going up the side of this. We were on Isla Isabela, which is the second largest volcano on Earth. And we were digging holes, collecting fungi to try to map where they live. And I was the first person to collect fungi in the Galapagos, this type of fungi. And we're on this volcano that's erupting. So there's like sulfuric fumes coming all around. And then we see someone coming by trying to catch their wild cow that got loose. And it just really was like, what is happening right now? Um, but the craziest thing that happened, I think, was I got parasites, which I collectively called Jennifer, like the group of them I called Jennifer, like these worms that I got from eating raw dolphin
Whoa.
in not Panama, in Ecuador.
Hmm.
So that was one of the weirdest, but many other odd things have happened. So like I've been near Siberia in Kazakhstan. So right across the border of Kazakhstan at the forest steppe is Russian Siberia.
Hmm.
And then it caught fire a few days after we left. And running out of water and having to only drink vodka in the field. It's been a crazy experience. I love all of the field work.
I've heard Kazakhstan's a pretty weird place.
It was a really unique place to do field work, and I hopefully am going to be going back this year.
Sure.
It's hard to get people excited about grasslands, but the steppe ecosystems are really charismatic to me. There are these really ancient grasslands where there's a lot of these fungi I study that live there. They love grasses. They live with most plant species, actually, about 70% of all plant species. But so it's like if you think of a grassland above ground, you just see little bits of grass, but there's many plant species even there. But below ground, it's like the Amazon rainforest. It is most carbon, most roots, most fungi are all below ground there. So it's really dense in grasslands. So we went to Kazakhstan to look at what Khrushchev had done to land there during the Virgin Land campaign. So it was this Soviet era campaign to rip up parts of the steppe and try to grow crops there. It ignored quite a lot of indigenous people who said this really won't work. That's why we're nomadic. We don't grow crops because they don't grow here. So this was the largest land altercation campaign in the world, and it just devastated the steppe. So we went and we started mapping what happened in these degraded grasslands to
Hmm.
to these fungi.
What happened?
Can't tell you, it's still being
Oh,
analyzed.
sure, sure.
Um, but for sure, I can say that ripping up native grasses and then trying to plant a non-native crop is not good for these fungi because they've co-evolved. These
Right,
organisms have co-evolved, right? Plants have co-evolved with their microbiomes, and so there's a question about, people always say like if you're going to plant a plant, use a native plant. But the question is, what is its native microbiome? Are these fungi
sure.
going to interact with the plant? Do they have this history together to recognize each other as well? So they
Right.
planted a lot of maize in the steppe, and maize
Hmm.
is not from Central Asia.
What does it look like to conserve fungi?
You even said the word yourself. So like it's historically been flora and fauna, and now we're trying to work to say add funga. That came from Juliana Furci, who is at the head of Fungi Foundation, one of our close partners. It's difficult. We're trying to figure out how can we make something unseen seen is the first thing. How do we make this visible for people? And so that's our first difficulty. So that's why we put a lot of time and effort on communicating the importance of these fungi. So that's like why I'm excited to be here. This information about what these organisms do is not to just sit in an academic lab. It's to explain that these are as vital as maybe the tree in your park near your house. The fungi underneath it are just as important. We put a lot of effort in visualizing these organisms. I think that is our most exciting thing. And in the time of climate change, they're providing at least me with a bit of hope that there are things we can do. So we didn't know not a few years ago, but we didn't know 10 years ago where these organisms really lived, and we're still mapping them. but we're now able to start making what I'm going to call precision planning. We can make with high, like with a scalpel, we can cut up parts of the earth and say, this is what we need to restore to add back infrastructure for below ground carbon entry. We haven't started doing that yet. We're still right now mapping the baselines of it. But I'm really excited about seeing what this could be in the future. Because once you know how much there is, like right now, you can start saying how much there could be if we restore it.
So it's only a small fraction of the world that's been mapped at all. But of all those sampling sites, are they kind of concentrated in one area? Are they distributed evenly across the globe? Or are there big holes in the map?
The samples of the Earth's underground ecosystems are heavily biased in space and time. So fungi sometimes go through moments in history where people are really excited about it, and then they stop and they get excited again. So the 80s were really big, and then the 90s a little less, and now we're back. And most sampling happens in the global north, which is very unfortunate. So the United States, Europe, parts of China are super heavily sampled for DNA of these fungi. We know very, very little about the other parts of the world. So, for example, in Madagascar, an island that is essentially its own, like it's massive. This massive island that's larger than many European countries, for example, has 50 samples. Yeah. And all of them only cover, I think, like 30 square kilometers of land. So most samples are not taken on Earth yet. The most exciting part about working with Spun is that we're trying to also decentralize this science. So we're not located in one country. We have scientists all around the world who are what we call underground explorers. They've applied for grants from Spun, and we have funded their research to answer their local question. And instead of doing helicopter science, we allocate the funds to them, let them answer their questions, or we also come and collaborate on some projects as well. But we have funded, I can't remember the exact number, but I think close to 100. I go to spun.earth to find the exact number, but I think close to 100 underground explorers now. We're sampling the most extreme ecosystems. Like these are the most badass scientists. Like I was in the Amazon rainforest and I forgot snakes were there and I went out screaming like, ah. But then these people run into the Amazon and collect samples and and there's beauty to it because they know everything about these ecosystems. So they're generating this knowledge and it feels really nice. It feels like we're moving a bit past the decolonial science and we're letting people answer questions. And it's at the end of the day, it's also the flow of resources from the global north to the global south, as opposed to resources in the global north being used to sample the global south.
That's so important. That's been such a deficit to conservation and to science in general historically. For people who are new to the idea, helicopter science is where researchers from a distant land, usually Europe or the US, drop into somewhere like the Amazon or like Madagascar or somewhere. They take a few samples, they collect a couple bugs, and then they fly back home and never really connect with the researchers or the people who are from the place that they're studying. There's a number of problems with this, which you could imagine. Some of them are kind of social justice type problems. Sometimes it reduces the opportunities from people in the local area. But even just from a purely self-interested science perspective, it's mistaken because if you think about it, the people who are going to be the best experts on a place are the people who are from that place, just by pure virtue of exposure. I went to the Amazon for a project in southern Peru a few years ago. I didn't know anything about what I was looking at. It was bizarre because I realized that I was totally blind to a lot of the things that were there. At one point, I'd been there for a few days and someone pointed out that there were these cicada burrows that make these chimneys that are about 6 inches tall. And that once they pointed them out to me, I saw them everywhere. They were covering the whole ground. I hadn't noticed them at all. One of the most common things on the forest floor. I just felt totally out of my element compared to the southeastern United States where I've spent most of my time. So these collaborations are with people from the place you're trying to study are really important to both the future of conservation, which requires collaboration with the people who live in the place, and also they're going to make us go a lot faster in terms of learning about these places and understanding how ecology works.
I just feel this so strongly. So we're working on another project that we're hopefully going to submit very soon. And we're just trying to make sure all of our science can be a bit more inclusive. So we've done this, we're doing this big, what we call a meta-analysis, where we read a bunch of other papers people have done and summarize it and reanalyze it in a big like global view. And for this, most people just read papers in English. But we have looked and I tried to identify papers in 11 different languages. And so we
Mhm.
read papers only in four of them because there was only four languages papers were available. But so I read the papers in Portuguese and Spanish. And that's where we got all of our samples from the Amazon, every single sample. So if people did this kind of English only or what often people think of as this Western type of science, you would have missed so much information. So there's ways to not do helicopter science in multiple ways. I think one is not physically going to a place without having a local collaborator who it's their question that's being answered. You're tagging along. But also those scientists who don't go out to the field, there's ways to do much more inclusive science from your desk.
I have a couple listener questions for you. Ocho wants to know about our common ancestry with fungi in general, the understanding they're actually closer related to us than they are to plants.
This is true. They are more closely related to us, but we have to go so far back in history. And to be honest, this is a part of fungal biology I do not remember super well. Like that's the lie. They tell you that like I'm doing a PhD in evolutionary biology working on the evolution of cooperation of fungi and plants,
Mhm.
but I've never had a class on fungi, like not
Right.
once. Like this is the lie. I can say they're much more similar to eukaryotic cells. It's much more similar to animals.
Mhm.
They don't have cell walls like plants do. Yeah, they don't have cell
Mhm.
walls. They have cell membranes. Don't quote me on this. I don't remember all of this, but they're more closely related to us. I'll add one thing to that. That's why the fact that fungi are more closely related to humans than they are to plants. That's why like fungal what would you call it? Like infections are so hard to treat because the medicines kill us too.
Court asks, what kind of memes do they share?
Oh my God, what kind of memes do they share? Well, I don't know if they are sharing them as more if like, they're sophisticated organisms. They're making the memes, but they
Mhm.
know that, social media can sometimes be a bit too much. So they really make memes, put them out, but they don't spend too much time on it. But I like to imagine that they all have like a little party underground and like for Halloween, they all have to dress as their favorite meme. Like, yeah, I don't know how much like do the people on the podcast, do you all know like the current memes? Like,
Yeah, yeah.
yeah, I think they would be like the one would come as like the cuckoo cuckoo woman. This might be way too niche what
I
I'm
don't
doing
know if
right
I
now.
know that one. Yeah, I was scared when I said that because it seems like there's so many that you can't keep track of all of them. But
I just went to a party and it was like come as your favorite niche internet meme, and I went as like Paul Mescal and his little shorts, but someone else went as Kamala Harris's coconut tree,
uh-huh.
finance. But yeah, they would be sharing a lot of memes, is I guess the short answer. A
Sure.
lot of memes.
You worked on the atmosphere as well, and bacteria moving in the atmosphere rather than fungi in the ground. What were you studying about that, and what did you find out?
So back at my start, I've been doing microbiology for about 10 years, and I was bacterial the whole time. I first did urbanization in Texas, and then I moved to cities. So I did Philadelphia, and what I did was kind of insane. I would put on what looked like a jetpack, and it was a vacuum, and I would walk through subway cars collecting atmospheric air. And then there'd be microbes on these little filters we would collect, and I would extract their DNA and look at them and try to correlate that to what times of the day people were riding the subway. But more crazy was our question was how does the 3D shape of the city relate to the diversity of organisms you find in the air? So we built these three-dimensional models of Philadelphia and we strapped this vacuum to the roof, a very fancy vacuum, to the roof of a car, and we drove these routes all around the city, starting and stopping, collecting these filters of bacteria. And so we then used a mixture of satellite imagery and DNA sequencing to correlate buildings with what's in the air. And so we're actually doing this in Utrecht, in a city a little bit south of the Netherlands right now with fungi.
How does it relate to fungi? Is it kind of the same method? You're mapping the spores in the air?
We were trying to collect where we were trying to do spores. So in Utrecht, they built green roofs. And I'm so excited about the idea of green roofs because it's like this new piece of green space in a city. It's really hard to get people to rip up a building and put a park there, but you can just put a green roof on top of it and boom, you have more green space. Utrecht put in 300 bus stops with green roofs on it. And so they added like, 1 square kilometer of park space by doing this. So it's like a whole new park in a
Wow.
city. We collected about on 30 of these roofs and then 10 parks around the city, fungi living inside of them, and are identifying the sizes of their spores to see if the size of their spores is related to the three-dimensional buildings around them and how tall they are, their shape, how complex they are. This can help us start to figure out dispersal patterns of how these fungi move through the air because their spores move through the air as well. And it's very generalizable. If we know things like that, we can even think about monitoring where fungi might actually start to establish new ecosystems. Or you could generalize it to things like the dispersal of like pathogenic spores in the air.
It's kind of like a volcano went off and they're recolonizing the new area as it progresses.
Yeah, that's kind of what we're looking at. It's cities are unique because in the city you really have like a lot of built environment, and then you have these patches of green space. And the question is, can these fungi disperse between patches, or are they very isolated? Cities are often much more diverse than actually the area around them. You can think of them as an evolutionary incubator for species, making them evolve new traits and new species to come out. But outside of cities, you have almost always agricultural land where they're ripping up the soil, tilling it, physically cutting mycorrhizal networks. But at the same time, the hypothesis is spores from those tilled soils get picked up in the air, disperse into the cities. And our question is, does the shape of the city influence that at all?
Do you think that the cities are going to eventually, or maybe now, function as a reservoir for biodiversity, at least with fungi?
I think they reservoir gives the idea that it was something historical. I think it's novel biodiversity you're finding in there. So they're novel
Hmm.
ecosystems in themselves.
So you mean like they're full of invasive species basically, rather than they're retaining stuff that was there before but got messed up around them?
A mixture of invasive species, but these species, Utrecht is a thousand years old. This city is a thousand years old. Evolution has happened for a thousand years. There's new species that have evolved there that would not have been there in the past.
Wow, that's cool. That's cool to think about.
Like I think there's like this thing about geckos evolved like stickier stickier like paws or fingers, what feet, whatever you call the part of the gecko I'm trying to talk about, in
Sure.
cities compared to outside of cities, so that they can climb up streetlights or something.
Yeah, I haven't heard that specific one. It wouldn't surprise me at all, though. In Florida, we have this crazy thing that's going on where there are a bunch of different anole species. Anoles are these little lizards.
Oh, it is this. Yes. Wait, I'm mixing. Wait, tell me. Yes, I think.
Okay, so in Florida we have one kind of anole. In the Caribbean there are a lot of different species. But in Florida we only had one. We had green anoles, which are they live on the ground and in trees. Historically, they're green. People here actually call them chameleons sometimes because they can change color. Not very closely related to chameleons, but they're just these little guys bebopping around. Something called a Cuban brown anole, it's another species, obviously from Cuba, got introduced into Florida over the last 100 years, and they don't go into trees very much. They don't go very high into the trees, and there was some concern that they were going to displace green anoles over time because it seems like, just anecdotally even, it just seems like a lot of the time when you see brown anoles, you don't see green anoles anymore. But they did this crazy project where Florida is surrounded by islands, by little coastal islands made out of sand dunes.
Is that the Keys?
The Keys are a little different because they have like a limestone base to them. They're kind of like real land, whereas these barrier islands, they're more like sand dunes. They are sand dunes, and they move naturally. Humans have kept them in one place because we built stuff on them, and we don't like it to move. But it's kind of like a big glacier made of sand. Um, so there's these huge dune islands all around Florida, and a lot of coastal places have these. And it set up a weird laboratory. And what they did is they took 6 of these islands that didn't have brown anoles on them, and they put brown anoles on 3 of them just to see what would happen. And what they found was that over the course of about 15 years, the islands which had brown anoles on them, the green anoles evolved different feet.
This is what I was remember. This is the yes. This I am mixing it up with geckos or something. I don't know animals. What happened to their feet?
So this would not surprise me at all if the gecko thing was also true. The thing that happened with green anoles is that they used to have, back some point in their evolutionary history, they used to be more tree based than they are now. So what actually happened is, we think, when they got to Florida, when their ancestors got to Florida and were the only anoles in the continental United States, they generalized. So they became the lizard that took up the ground and the trees. But in a lot of places where there are anoles, there's different anoles that take up different specializations. So some of them live on the ground, some of them live in trees. They have divided up the space, and some of them are better at different things because when they're next to each other, they're too closely related and too similar, and they wind up conflicting. So what happened on these islands is that in the space of 15 years, the green anoles didn't die out. What happened is they got displaced into the trees, and the brown anoles took the ground. So the green anoles, apparently, the craziest thing is that their feet change shape so fast. Normally, you'd expect, something to evolve over the course of like thousands or millions of years. But maybe because they had this foot shape in the past, it just took a smaller mutation to kind of revert their feet to where they were before. That part, I'm not sure how much of that is just the story we're telling and how much of that we know exactly. That gene definitely did get that what single mutation happened to change their feet. It's allowed them to just divide up these niches. So a lot of the places with brown anoles, and it happened independently on multiple islands, the same mutation happened. So a lot of places, the thinking is that they're not actually getting pushed out, they're getting pushed up, and they're just adapting to this two anole or more environment that they haven't been in since they got to the mainland, some time ago.
This is like such a perfect example of why city or urban areas or human modified ecosystems are these like evolutionary incubators. It sounds like it relied on the introduction of this different anole that humans brought. Mm-hmm. Like you just told a beautiful story. I can't wait till we can do that for fungi yet. We know so little, we can't even study them to like that extent.
What a crazy world. There's so much stuff to look at. That was about all the questions I have for you. Do you have anything in particular that you want people to know about fungi or about your work?
I don't know where this fits, but like I've talked about these fungi and these maps we make of where all the species, how many species of fungi live on Earth for every square kilometer of Earth, how much hyphae there are. But at the end of the day, all of this is based off of the information that we have, and we're getting new information every day. So these maps and these understandings of fungi are these like living documents that keep getting updated. And at the end of the day, the thing I'm most excited about is about where we're wrong. Actually, is quantifying our uncertainty and using these quantified uncertainties as treasure maps to know what to do next, and where to go sample, who to work with, and what ecosystems are at threat. So I sound very confident in everything I'm saying, but that's because we look at where we're wrong and we try to identify where we're wrong with every single step.
Looking for holes in the map.
Literally, I think of it like I have this one map that it's colored red, it's black to red, and red is like priority, where we know the least amount of information about fungi. For example, we don't have a single sample in the Congo.
Wow.
Yeah. And that's an area that's larger than most countries in Europe.
Crazy.
Yeah.
Why not? Is it hard to mail stuff from there, or is it just you haven't gotten to it yet?
Don't get me into the mailing. Oh my God. So when I was in the Galapagos and going island to island, I had to stop at every single island to like take everything out, get it checked, bring it back. Mailing things is super difficult. I recently DM'd on LinkedIn the Minister of the Interior for Madagascar to be like, hello, how do I get a permit to mail DNA out of your country? Because they don't have, a lot of countries don't have things set up for this, or it's illegal to mail DNA. It's very difficult to do this.
I could keep asking you questions for hours about this. This is just such a crazy field. Where can people go if they want to follow your stuff?
I would say probably follow Spun on Instagram. Other than that, I'm on Blue Sky as the Crow. Yeah, I would say do that. Go to the spun.earth website. Just look out for us. Like the fungi are everywhere. Like we'll find you. We'll grow our network and find you.
Well, that's our show. Thanks for listening. You can support the show by going to patreon.com/wildgreenmemes. That's memes with an M. We send out stickers to anyone who is our patron at the possum or the shark level. Possums get 1 sticker a month. Sharks get 2. Until next time, thanks for listening. I'm Rhett.
And I'm Justin.
And happy Year of the Raccoon.